Well, when one talk about prophylactic HCV vaccine trial, I can't think of anybody else more appropriate to talk about it than Dr. Andrea Cox, who together with Kim Page really ran the first randomized controlled trial for HCV vaccine. And Andrea is a professor of medicine and oncology at Johns Hopkins School of Medicine. As you heard from Dr. Thomas, Andrea is part of this highly productive team that made important contribution to HCV. She in particular is an immunologist, so has really contributed to vastly to the knowledge of immune biology of hepatitis C. So without further ado, I'll let Andrea tell you about her experience of running these large trials and what some of the insights she had learned from this trial. Andrea?
Thanks very much, Jake, for that kind introduction, and John and Rajan and Chris Walker and others for organizing this fantastic symposium. I'm going to talk a little bit about some things that we have learned subsequently on the trial that I hope will be informative and provide some insight into mechanisms of the trial. And I'm sorry, I'm just trying to get my screen. Is that showing now as the full screen? Yeah, yes. Okay. Mm-hmm. So I'm really giving this talk on behalf of a group of individuals who've helped us to further understand some of the factors that may have made the first prophylactic HCV vaccine trial less effective at preventing chronic infection than we hoped. And so this trial was called Vaccine as Prevention, or VIP. There's the clinicaltrials.gov designation number. And the design of this was really to take HCV uninfected, so antibody and RNA negative people who inject drugs aged 18 to 45, both men and women. And this was really a Herculean effort on the part of great teams at Johns Hopkins, the University of California, San Francisco, and the University of New Mexico. And in this trial, we first looked at safety parameters, and then with good safety data progressed to a second stage that involved safety, immunogenicity, and efficacy. And this was a double-blinded, randomized, placebo-controlled study in which individuals were randomized 1-to-1 to receive placebo or vaccine. And we also stratified based on some of the comments that were made by Dr. Hufnagel and Dr. Thomas on sex, and also on lambda interferon status to make sure that those were balanced in the placebo and the vaccine group. And this really was a strategy based on beautiful work done by Alfredo Nicosia, Antonella Folgori, Stefania Capone, and others, showing that this vaccine strategy of using a chimpanzee adenovirus 3' followed by a modified vaccinia Ankara boost 8 weeks later, and that those vectors encoded genotype 1b non-structural proteins. That vaccine strategy was subsequently shown in healthy volunteers not at risk for hepatitis C virus infection by Ellie Barnes and Paul Klinnerman in the study I show here at the bottom, really to mount excellent T cell responses, and therefore to really demonstrate some promise in terms of potentially offering protection against chronic hepatitis C. So with those safety and immunogenicity data in healthy volunteers, we proceeded to enroll individuals at risk through injection drug use for hepatitis C. The results of that trial were published in the New England Journal in February of 2021, and the reference is there. But what the goal was, in addition to assessing safety and immunogenicity, was to determine if there was efficacy in preventing chronic HCV infection. As Dr. Thomas mentioned, the disease from hepatitis C almost universally results in persistent infection. And so the goal was really to prevent individuals from becoming chronically infected, to skew them towards the individuals who cleared infection, as we see in about a quarter of people who are spontaneously or naturally infected in resolving their infection. Unfortunately, that was not what was observed. And in this trial, you can see here, both in the per protocol and modified intention to treat, really no significant difference between those who received vaccine and those who received placebo in the development after infection of chronic infection. And so the primary endpoint was not met without a reduction in progression to chronic infection. And this was, however, accompanied by a suppression of peak HCV RNA levels in vaccinees. So you can see here the curve. This is the geometric mean of HCV RNA in individuals who fell within the placebo arm in the black outline, or in the vaccine arm shown in gray. And this is actually a significant blunting of the peak of HCV RNA. So what you see is an upswing in viremia from incident infection to month 1 in those who received placebo. In contrast, in vaccine recipients, on average, there was a substantial blunting in that upswing in viremia did not occur. I will say that this is an average, and there were individuals who were vaccinated who actually looked like placebo recipients with that big upward swing in HCV RNA. And one of the things that our U19 is focused on is investigating what might be different in the individuals who managed to suppress peak viremia compared to those that did not. But nonetheless, and I will say that this is promising in that, in contrast to HIV vaccine trials that really didn't show differences in HIV RNA levels in vaccinated individuals, this does suggest that the T cells generated by this vaccine, and again, there's no envelope in the vaccine, so this is not generating neutralizing antibodies, but the T cells generated by this vaccine did actually manage to suppress and really press on the virus in some way, again, suggesting some ability of an immune response from vaccination to create pressure on the virus. So part of the goal of the current U19 with Ellie Barnes, Paul Kleinerman, Andrew Fliak, Justin Bailey, and Stuart Ray, part of our goal is really to, and Georg Lauer, is to understand how to enhance the ability of T cells generated through vaccination to basically truncate and terminate viremia in vaccinated individuals. So to summarize this trial, which again is published, so I'm not going to dwell on it substantially on the results, except to say there was no problematic safety signal from this vaccine. Again, there was no significant difference in progression to chronicity, but there was the suppression of virus transiently, suggesting immunologic pressure. And one thing that we noticed was, while this vaccine was immunogenic, it was much less so than the healthy volunteer trial, including the trial done by Paul Kleinerman and Ellie Barnes. So I'm just going to detail that comparison a bit more here. So in our trial, 78% of vaccinated individuals crossed the threshold of positive. They mounted a T cell response to vaccine antigens. And that was in contrast to 100%. So everyone in the healthy volunteer trial mounted responses to vaccination. And I will say that the vaccine trial conducted at Oxford was done the analysis was done using fresh cells. Ours was done using frozen cells. And when you take that into consideration, there was still about a 3-fold lower magnitude of T cell responses in the VIP trial compared to the healthy volunteers by ELISpot assay. And so, and we are looking in much greater detail at the T cell responses induced, characterizing them extensively in the current U19, but we postulated that there were a few things that could explain this decreased magnitude of T cell responses observed in people injecting drugs in the VIP trial compared to those not at risk for HCV infection. And the first was, as you may know, one of the things that people consider when choosing a vector for a vaccine is a preexistence of immunity to the vector, because in the setting of immunity to the vector, you can see diminished immune responses to the antigen carried by the vector. And that is highlighted really in the Merck STEP trial of an HIV vaccine. And basically, on this, you can see responses to antigen. If you focus on this bar that says at least, one antigen of HIV being recognized following vaccination, and they stratified individuals by having antibodies. In this case, it was Ad5 that was a vector, not the chimpanzee adenovirus 3 we use for prime. but they stratified by undetectable and detectable, as well as titers, neutralizing antibody titers less than 200 or equal to 200 or greater than 200. And focusing here, you can see the difference in individuals with with anti-Ad5 neutralizing antibody titers at or below 200. 86% of them responded to vaccine with a response to at least one antigen. And that compares to 68% in those who had higher titer neutralizing Ad5 antibodies. And that trend continues as you go down the graph and look at the number that recognize 3 antigens. It's 58% versus 34%. So this really established, as has some other trials, that having neutralizing antibody titers against adenoviral vectors in excess of 200 is associated with diminished responses to vaccination. And so the question we asked, and this was both the data and the slide are courtesy of Stefania Capone, who's an expert in assessing neutralizing antibody titers against ChAd3. And what we can see is actually a comparison between this is the 200 cutoff I mentioned shown in red. These are neutralizing ChAd3 antibody titers shown in our trial, where about 20% of individuals in the trial had ChAd3 neutralizing antibody titers at or above 200. And really our concern that this population of people who inject drugs might be more heavily exposed to adenoviruses and have more cross-reactive higher cross-reactive antibody titers was not borne out. If you look at the percentage shown along the bottom, and Ebola and other trials, as well as a healthy volunteer population, if anything, the percentage of individuals with anti-ChAd3 neutralizing antibody titers in excess of 200 was the same or lower than in populations and in other populations. And in these populations, for example, in the Ebola trial, again, the immune responses were much more robust. The T cell responses were more robust than what we're seeing in this trial. So it does not look like the existence of high ChAd3 neutralizing antibody titers at baseline explains this diminished response in vaccine participants. So this is really illustrated that here, though, we can say that despite the fact that it didn't have higher than average ChAd3 neutralizing antibody titers, there is an effect of the existence of ChAd3 neutralizing titers. So this is stratifying on the top graph by individuals who at baseline had ChAd3 antibody titers above the lower limit of detection, and on the bottom, those with ChAd3 antibodies below the lower limit of detection. And if we focus first on the bottom graph, you can see that in response to the adenovirus prime, there's an increase in the spot forming cells per million PBMCs. So you see a bump in the graph that reflects this priming of the response. That increase in T cell responses as measured by ELISpot assay is not present in those with antibodies against ChAd3, neutralizing antibodies above the lower limit of detection. And you can also see that the boosting of occurs with the second peak in T cell responses, that it's also blunted in those with ChAd3 neutralizing antibodies compared to those who do not have this. But I will also point out that the response in those without any neutralizing antibody directed at ChAd3 is still quite low. So in the lower graph, you still see a pretty minimal response in terms of the number of T cells generated. So this is simply not the only explanation for the reduced response in these individuals. While having neutralizing antibodies against ChAd3 had an effect, even those without any detectable neutralizing antibodies had blunted responses compared to individuals receiving vaccines, in fact, the exact same vaccine regimen in other trials, including Ellie Barnes and Paul Klinnerman's healthy volunteer trial. So what about this limit of 200 as the neutralizing antibody cutoff at which anti-adenoviral vector antibodies become a problem? When we look at the effects of anti-neutralizing antibody titers based on the actual titer, If I divide individuals into 18 to 100 as the CHAD3 neutralizing antibody titer, that's shown in black, or in blue, individuals who have CHAD3 neutralizing antibody titers greater than 101, you can see that this is where the prime occurred, and then 8 weeks later was a boost, or visit 6. Visit 8 here is the peak of the response, which is a week following that MBA boost. And you can see significant increases in the ELISpot response in individuals at that peak, as well as at other time points in individuals with anti-CHAD titers 18 to 100. When you get over to 100, actually, there's still a significant increase in individuals at visit 8, but that difference is actually not present at later time points. And if we choose a higher cutoff, which is over 150, we basically see no significant difference in any individual between baseline and later time points at the ELISpot response. So essentially, in no individuals with anti-CHAD titers over 150 were any significant ELISpot responses induced in terms of being different from baseline. And while you see a trend towards an increase in the green group, it's not a significant increase. And so the effect of these antibodies is mediated at a lower anti-CHAD3 titer than has been seen in previous trials. And rather than 200 or more being a problem, the problem in suppression of immune responses to vaccination begins in this patient population, in this study group, at titers that are somewhere above 100 and really clearly problematic by the time you see titers at 150. So at a lower level of anti-CHAD3 antibody, neutralizing antibody titer, we see impairment of the T cell responses induced relative to other trials using adenoviral vectors, including this adenoviral vector. So anti-CHAD3 is definitely an issue, but I mentioned that individuals who have undetectable neutralizing antibodies against CHAD3 also had blunted responses, which caused us to postulate that potentially drug use or nutritional status impairment in individuals who are injecting drugs may impair immune responses. There are data for this in the literature, and that these two trials I reference at the bottom are HIV vaccine trials. And in these HIV vaccine trials, they compared immune responses in individuals vaccinated because they were at risk of HIV through injection drug use to individuals vaccinated against HIV because they were at risk because of sexual exposures. And they actually found significantly blunted responses to the vaccine in people who are injecting drugs compared to those who are at risk from sexual behavior. So this lends support to this, and this is something we are investigating, including Niket Chowdhury and Kim Page are working on determining if, for example, a specific drug of abuse is associated with lower immune responses to the vaccine. So I just want to highlight this paper that's not yet peer-reviewed, but has been published in medRxiv by Rafiq Sekali's group. And these were looking at drivers of immune response to dengue vaccination. And I will point out that they were looking at drivers of antibody responses, not T cell responses. But in this systems immunology approach, they actually found that certain cytokines, when high, were associated with development of a narrow breadth of antibodies, as opposed to high TGF-beta signaling and active immune cell metabolism, particularly monocyte metabolism, led to a broader neutralizing antibody. So this gave us the idea that we might consider the immunometabolic state of these individuals, which we are investigating, but also that we could simply look at cytokines at the baseline at the time of vaccination to see if we saw any differences in baseline cytokines in these individuals. And actually, we then stratified based on the results on the interferon gamma ELISpot assay, the individuals into the top responders and those who did not respond at all to vaccination, and across a series of cytokines and chemokines. And essentially, it's a little hard to see the average of each, the median, excuse me, is shown in the black bars. But the summary is that there were actually no significant differences in the baseline cytokine or chemokine levels, and a whole host of cytokines and chemokines assessed in those who responded robustly to the vaccine compared to those who didn't respond at all. So this does not seem to outline to us a difference in the patient population at baseline or in the participant population. So Barbara Rearman has data that repeated low-level exposure to hepatitis C virus can actually blunt immunity to the virus. And we thought that maybe these individuals who are repeatedly exposing themselves to HCV, that this may in fact be the explanation. All of these individuals were screened at enrollment for T-cell responses to HCV, as well as antibody to HCV, and no one who had either T-cell or antibody responses to HCV at baseline was enrolled. So we knew at baseline these individuals did not have at least any immunologic evidence of exposure to the virus. And we then looked, this was work done by Kim Rousseau in my lab, but we then looked at the end of the trial. And at the end of the trial, remember, in vaccinated individuals, we can't look for T-cell responses to the non-structural proteins because they're induced by vaccination. But we looked at both antibody and T-cell responses to the envelope and core proteins of the virus that were not in the vaccine. And again, as mentioned at the start, but also at the end of the trial, we found no evidence of immunologic evidence, at least of exposure to hepatitis C, that might have explained that even though we didn't have detectable viremia in these individuals, they had low-level responses. So this is looking at individuals who did not become infected. And there was no evidence of responses to immune responses to the antigens not in the vaccine. So no evidence of this, at least by immunologic assessment. So I think that while we don't understand what led to this profoundly lower immunogenicity of this exact same vaccine in the PWIDs compared to healthy volunteers, I think this argues for universal vaccination. Vaccinating people when they're not at risk for through injection is potentially a way to harness immune responses that will be more robust than once individuals are injecting. Because again, even the anti-ChAd3 data suggests that there's something about these individuals that made them less responsive, and the ChAd3 antibodies have an effect at lower levels. And so we just think that it would be one more reason, and I will say one more reason, because finding individuals, as Dave Thomas mentioned in his talk, at risk for HCV who are not already infected is also quite challenging. So I do think, as with the other hepatitis virus vaccines, that potentially universal vaccination is a strategy to mitigate this. But I also think it highlights the importance of testing vaccine candidates in people who inject drugs, because the results may be, as illustrated in this vaccine, quite different compared to individuals who are healthy and not at risk for Hep C. So to summarize, the T-cell responses induced were considerably less robust in healthy volunteers. Baseline high titer ChAd3 neutralizing antibodies were not present at a higher percentage in these individuals than in other trial populations where responses were much more robust. The effect of ChAd3 neutralizing antibodies occurred at lower neutralizing antibody titers in this study than in other studies. We found no evidence, at least immunologically, of low-level HCV exposure. Remember, everyone in this trial was being screened by viremia as well. So there's no evidence of exposure by viremia either. And I think we really need to do ongoing research on the potential differences in the immune response in individuals, people who inject drugs, and we are certainly doing so. I do want to highlight, though, that the suppression, even with not particularly robust T-cell responses, the suppression of viremia seen in this trial, to me, suggests that, again, it is possible to mediate immune pressure on the virus, and I'm optimistic that we can do so to affect a different outcome with future vaccines. And with that, I just want to acknowledge the individuals. Again, there's a whole host of people who participated in the vaccine trial, and they're highlighted in the New England Journal paper. So I've really listed the people here who have helped to conduct the work I showed today. And we're always grateful to the study subjects who are willing to participate in this trial to help us learn more about how to prevent hepatitis C. And with that, I'd be happy to take any questions.
Okay, thank you, Andrea. You certainly have shown some new data. I haven't seen it before, so quite impressive. I see a question in chat by Belly Polendron, and he asked, could the reduced immunogenicity of the Chat3 vector be due to pre-existing antiviral innate immunity, such as incorrect expression IgGs and because of the prior exposure to some other viral infections? Obviously, these PWID are frequently exposed to many different types of viruses.
I think that's a great question. And Abraham Condathil has done a really beautiful job looking at the virome in individuals who are injecting, and it is unique. So actually, individuals who inject drugs are exposed, as noted, to a variety of viruses that are not necessarily as commonly sources of exposure in people not injecting, which is one of the reasons we hypothesize they might be exposed to viruses that had some cross-reactivity to the Chad3, and we might see higher titer neutralizing antibodies there, which again we did not see. So we have PBMCs from baseline in these individuals, and we are really querying the baseline profiles of the cells. And we started with plasma, but we'll be looking at the cells to see if there are immunometabolic differences, which is something our lab has done extensively in SARS-CoV-2 acute and following vaccination, for example. And so we will be looking to see if we can find differences in the at least the peripheral blood mononuclear cells at baseline that may explain differences in their propensity to innate signaling or to yield downstream adaptive immune responses. But I think that's entirely possible that their prior exposure to a number of different viruses may alter the subsequent immune response to many things and not just HCV.
Okay, great. There are a couple of questions in the Q&A. You answered the first question by Anna Chumbin in terms of cross exposure to other viruses. And there was a second question about whether you could explain the difference by genetics, viral genetics between the vaccine and placebo groups.
Yeah, so the host genetics, we did our best to control for that. We stratified by lambda interferon and sex and the randomization was good. There were no issues in the trial. The age, BMI, all of the things, the randomization was good. So host factors didn't seem to be different between placebo and vaccine recipients. In terms of the virus, I mean, I can answer a couple of questions I've seen flash by, but we are sequencing incoming, the infecting viral strain, looking for differences in the infecting viral strain compared to vaccine antigen, which was the BK, genotype 1b BK virus, and looking for cross-reactivity of vaccine-induced. This is work that Georg Lauer and I are doing with our teams in our U19, looking at cross-reactivity of the T cells induced by vaccination at incoming virus, and where there is cross-reactivity, and I think the suppression of peak viremia suggests there must be some, where there is cross-reactivity, whether there's escape in the subsequent escape in the subsequent T cell epitopes targeted, which Chris Walker, Barbara Rehrman, and our group and others have shown. So, yeah. Let me see what else.
Andrea,
we've got a lot of questions.
Maybe you can just answer 2 more questions, then
Sure.
we have to move on. You can pick them.
Oh, okay. Well, I see like 17. It might be hard to, yeah.
Well,
we're
maybe just the first 2, yeah.
In terms of Tregs, we are characterizing the cells at baseline, again, metabolically, as well as profiling the cells present. This is something we did in acute SARS-CoV-2 infection, and we're essentially completing the exact same immunometabolic profiling, which tells you not just what cell types are present in the peripheral blood in abundance at baseline, but also what their metabolic state is to see if that predicts differences in response. It is not, I will tell you that it is not a matter of the robustness of the response. There were individuals who suppressed peak viremia who did not have the most robust responses, and others with excellent T cell responses who failed to suppress initial peak viremia. So it's complicated, and we hope that some systems immunology, as well as this novel immunometabolic profiling that we're doing, will help us to answer some of those questions. I think that covers a couple of the questions. Of the questions.
Yeah, I do see that Michael Houghton like to answer one of these questions live. This is a question on side effects reported by both PWID and healthy volunteers in these trials. So I can't really reject Michael. So Michael, go ahead. You can answer live if you wish.
Yeah, sorry, Jake. That was a mistake in my typing.
Okay.
I did have a question in chat asking Andrea, are you looking at viral escape mutations from CD8 positive T cells as an explanation for the lack of efficacy?
Yes, so we're sequencing incoming virus, looking where it differs from a vaccine strain, and where there's cross-reactivity, we are looking for subsequent escape as an explanation, because this suppression of peak viremia did not result in higher rates of control. So I think it's an excellent suggestion that there may simply be escape from the T cell responses that are directed at cross-reactive antigens.
Yeah, because the Akeros vaccine had a huge number of epitopes, CD4 and CD8, and many of those were variant. And Chris Walker has shown very well in the chimp model that you can readily get escape from CD8 T cell responses, obviously at variant epitopes that are not highly conserved. So
yeah, we see the same thing in humans infected with HCV. Stuart Ray and I published a paper in 2005 in JX Med highlighting that actually the 69% of T cell epitopes targeted in the acute phase by those to go on to persistent infection undergo substitutions that negate recognition by the T cells present in those individuals. In contrast, individuals who control HCV, none of them had substitutions within T cell epitopes recognized by T cells generated in response to infection. The question is that a cause or a consequence? Is escape mediated by persistent viremia, which is going to happen anyway, or is it the cause of persistence? And we are investigating that, as well as looking at escape from neutralizing antibody responses with Justin Bailey and Andrew Fliak in our current U19.
yeah, thank you.
Okay, well, great. Thank you very much.